Hello to everyone. Thank you for joining me for this talk where I shall describe a novel probiotic concept with its implications for the modulation of the immune system and for the gut and the brain microbiota. Essentially the title of a paper that we recently published in the American Journal of Immunology. Before I start, of course, I wish to thank you all for attending this webinar and for watching this video. And of course, I also wish to thank Stefania and Fulvia for their priceless support. Without their help, there wouldn't be anything of the things that I shall describe today. Before we start, of course, an important disclaimer. We are not making any medical claim. The contents of this presentation are only for informational purposes. They are not intended to be substitute for professional medical advice, diagnosis, or treatment. And of course, this presentation does not provide medical advice, diagnosis, or treatment. Please always seek the advice of your physician or other qualified health provider with any questions you may have regarding a medical condition. And also, please consider that the results and the considerations that will be reported in this talk have been observed in different countries in the world. Therefore, I suggest that you check with your national or state health authorities for the current legal situation concerning the approaches that will be described in this talk. And always remember that complementary approaches, such as those described in this talk, are indeed complementary. They are not alternative to the standard of care recognized and approved by competent health authorities. I have to disclose my conflict of interest that is rather apparent. I am the inventor, together with my wife and lifelong scientific collaborator, Dr. Stefania Pacini, of Bravo and its derivatives. I'm also the founder and the CEO of the Silver Spring company, the Swiss company that produces and distributes the Bravo. I'm also the inventor of the Rerum, a powerful immune stimulant, and I collaborate with the Sophia Health Institute of Seattle, Washington State, together with Dr. Dietrich Klingart. Now, let's start for real, talking about the most interesting things that we have learned in the past year or so. And they are the importance of the brain microbiota, the microbes inside our brains, and how we can reconstitute it. And also the role of the immune system in connecting the gut and the brain microbiota. Well, you may have had the opportunity to listen to the talks of the experts in microbiome medicine gathered at this summit, where many doctors have been talking about the microbiome medicine, how it could improve your health, longevity, vitality, and assist with unresolved problems. And I hope that you will have enjoyed, or you will enjoy, my talk, as well as the talk of Dr. John Gray, talking about brain function, mood, anxiety, depression, GI illnesses, Crohn's colitis, GI brain syndromes, and autoimmune diseases. I may have been the first to coin the words brain microbiota and how it relates to neurological diseases in a paper published in PubMed. Nevertheless, the presence of microbes in the brain is not my discovery, since it has been described by Canadian researchers since 2013 in this paper published in PLOS ONE. This paper on the left, published in November of 2016, is my paper where for the very first time the two words brain microbiota and neurological diseases are put together. Now, this paper published by the Canadian researchers is of particular importance because for the very first time it describes the presence of microbes inside our brains. Let's read it together. In an organ, that is the brain, widely assumed to be free of infectious agents in the absence of a specific disease process, that is, in the absence of an infection such as meningitis or encephalitis, autopsied and surgically derived human brain specimens showed a restricted but distinct bacterial population in the present studies, which was composed of bacterial classes chiefly recognized in the physical environment, that is soil and water. In other words, inside our heads, together with our neurons and glial cells of the brain, there are microbes, and these are the same microbes that you find in the environment, in soil and water. How do they arrive, these microbes, to the brain? Well, they arrive carried by cells of the immune system that are activated lymphocytes and macrophages. And please remember the macrophages because they will come back in this talk later on. Now, quite obviously, the presence of these microbes inside our brains is of extreme importance. And as these Canadian researchers write, their capacity, that is the capacity of the microbes for influencing brain function, is immense. And immense, of course, is a superlative. There is nothing more than immense. Therefore, we may say that the function of our brains, as well as the function of our minds, is dependent on the right balance of microbes that inhabit our brains. Now, there is something more. According to these Canadian researchers, the microbes may be responsible also for the evolution of the brain as we know it today, since we find microbes only in primates. We don't find them in dogs and cats, but only in primates and in humans, of course. It is interesting to notice that in 2015, we published 4 papers where we describe the role of a number of genes in the evolution of the human brain. These are 2 examples of 2 papers that we published in this context. But most interesting is this gene called RUNX2. RUNX2 stands for RUNT-related transcription factor 2. And in 2015, we described the role of this gene in the evolution of the human brain. Now, it is interesting to notice that this gene is involved also in conditions as diverse as cancer and autism. And the following slide shows our papers on this topic and 2 examples of papers describing the role of RUNX2 in cancer, in this case, cancer of the breast, and autism. On the left of the slide, you will see the 2 papers of ours describing the role of RUNX2 in the evolution of the human brain. On the right, you see this paper describing the role of this gene in autism, and here on the right bottom, you will find one of the many papers describing the role of this gene in breast cancer. Now, you may say, why is this gene so interesting? Well, it is interesting in this context because the expression of this gene is regulated by probiotics, as it was demonstrated by Italian researchers in 2013. In other words, whether this gene functions or not and how much does it function is regulated, it is dependent on probiotics. I'm sure that you are aware that fermented foods, that today we call probiotics, represent the first processed food consumed by humans. And therefore, it can be postulated that the brain microbiota, and therefore the evolution of the human brain, are associated with the consumption of probiotics by our ancestors. The following slide shows the paper demonstrating that a particular probiotic that is highly presented in our formulas, the Lactobacillus rhamnosus, stimulates the expression of RUNX2, or in other words, switches the gene on. This is the paper published in PLOS ONE in December 2013. This is the Lactobacillus rhamnosus inducing the expression or switching the gene on, if you prefer, of RUNX2. Now, it is based on these scientific observations that we developed a novel probiotic concepts, a series of concepts related to probiotics aiming at modulating the immune system and at reconstituting the gut and the brain microbiota. All these concepts are highlighted in this original research paper published in the American Journal of Immunology that you can download for free. I will show you later how. Our novel probiotic is more than simply a probiotic. It is a concept that represents an evolution of the concept that we first published in 2011 when for the very first time we reported its effects on the immune system, in those days on these cells called MALT, that stands for mucosa-associated lymphoid tissue, mainly lymphocytes and macrophages, as we report in this paper published in 2011. In the past 7 years, this concept of ours that nowadays goes by the name of Bravo has been popularized in a number of publications and books, in addition to being mentioned in several peer-reviewed papers that you can find in the major scientific databases such as PubMed of the National Library of Medicine of the US, Scopus, and Loop. For example, this newsletter from the Riordan Clinic of Wichita, Kansas mentions our concept as the super probiotic in 2013. And the late Dr. Jeff Bradstreet, a good friend of ours, in his blog published this post where he wrote that Bravo Probiotic actually is a system for making a new ecosystem, not merely a probiotic. And in the paper that follows, I published also a book together with Peter Greenlaw and Drew Greenlaw called Your Third Brain, where this concept is further explained, as well as in an interview that I gave in February-March 2017, when this interview has been published in 1,000 letters and it has been declared the most remarkable ever published by this magazine. Also, if you don't want to listen to my interview or read my paper, I encourage you to read Dr. John Gray's books that are international bestsellers. You may know that Dr. John Gray is the author of Men Are From Mars, Women Are From Venus of about 20 years ago, and very recently, in January of this year, 2017, he published another book entitled Beyond Mars and Venus, that is a sort of a sequel 20 years later of his first famous book, and we are honored that at page 167, he mentions the Bravo probiotic yogurt, the Swiss-made Bravo yogurt with 42 strains of probiotics that can, if eaten over 3 months, help restore the gut microbiome to dramatically improve digestion and support optimal brain function, again highlighting the link between the gut microbiota and the brain function. Today we know the brain microbiota. Should you be interested in something written in Spanish, there are a couple of more articles and interviews in Spanish in magazines published in Spain and distributed all over the world. Now, this novel probiotic of ours is based on a proprietary array of microbes that do not require dairies for fermentation. So here we see a major difference with the old Bravo. The old classical Bravo, that still gives us enormous satisfaction, is based on the fermentation of milk and bovine colostrum. This novel probiotic is based on microbes that do not require milk or colostrum. Now, each one of the many microbial strains that constitute this non-dairy probiotic would deserve a specific discussion that may take hours and I guess would become very boring. Because of this, here I shall focus on 2 strains only. One strain of microbes belonging to the prokaryotic domain, which means bacteria, and another one belonging to the eukaryotic domain. We as humans are made of eukaryotic cells, and there exist eukaryotic probiotics that help us with our health. The first strain I will be talking about is called Bifidobacteria or Bifidobacterium singular. And it is very well known that the homeostasis, which means the balance of life of Bifidobacteria, is typically altered in a number of chronic conditions ranging from autism to cancer. And such a lower relative abundance has been associated with systemic inflammation, which means inflammations in most organs and systems of our body, as well as with leaky gut. The following slides show 2 of the very many studies describing the positive role that is exerted by bifidobacteria in cancer and in autism. The first paper deals with colorectal cancer prevention thanks to lactobacilli and bifidobacteria, that again are strains highly represented in our formulas. And these healthy microbes, they help us preventing colorectal cancer through a number of mechanisms, which include reduction of DNA damage and, most important, modulation of the immune system. As they write here, immunomodulatory role of probiotics in colorectal cancer, and in particular, the ability to modulate, to reduce intestinal inflammation. That is a very well-known risk for colorectal cancer. And moving to another area of research, that is autism, in this paper published in 2015, dealing with the risks for developing autism, the authors working at the University of Delaware write, "It has been hypothesized that gut bacteria may contribute to the development of autism, and specifically, the relative balances between the inflammatory microbes Clostridium and Desulfovibrio and the anti-inflammatory microbe Bifidobacteria may become destabilized prior to autism development. In other words, too many inflammatory microbes, too little anti-inflammatory microbes such as Bifidobacteria. And this imbalance leads to a leaky gut characterized by a more porous epithelial membrane, resulting in microbial toxins released into the blood, which may contribute to brain inflammation and autism development. In other words, too little bifidobacteria, too much inflammation, too much leaky gut, too many toxins into the blood, too much inflammation in the brain. Now, the other microbe that I want to mention here goes for a very strange name, Cluiveromyces marxianus. Nothing to do with the planet Mars, nothing to do with Karl Marx, the philosopher, neither with the Marx brothers. It takes its name from another Marx, a biologist, a scientist who first identified this eukaryotic microbe. Again, eukaryotes are cells identical in principle to the cells of our bodies. Now, this particular probiotic, the Cluiveromyces marxianus, has a number of healthy properties, because it is known that it decreases the production of pro-inflammatory cytokines in human macrophages. In other words, it stimulates macrophages without inducing inflammation, and also modulates the function of a subtype of macrophages extremely important in health, that are called dendritic cells. In addition, it shows anti-inflammatory and antioxidative stress properties, antioxidant properties in models in vivo. And it favors the growth and colonization of the human colon by the bifidobacteria. In other words, it cooperates with the bifidobacteria, and the two strains together have a synergistic effect. And it increases the production of short-chain fatty acids that include acetate. Now, this becomes very interesting because it has been known since 1940, many decades ago, that acetate is of utmost importance for the establishment of a natural nutritional ketosis. That is what you try to achieve with a ketogenic diet. The nutritional ketosis that is known to have a number of therapeutic uses in a wide range of conditions. I'm sure that you are very well aware how important is nutritional ketosis in cancer, for example, but also in Alzheimer's, other neurodegenerative conditions, as well as in autism. Now, it is interesting that thanks to the Cluiveromyces marxianus, if you wish to achieve nutritional ketosis, you don't need any longer to starve, and you don't need to gulp down exogenous ketones. You simply have to let the microbe work for you, and you will naturally produce your own ketones in your body in the most physiological way. In other words, you will achieve all the benefits that are associated with a ketogenic diet without side effects. And the benefits of the nutritional ketosis are so many, in so many different conditions, that as of today, there are more than 2,000 peer-reviewed papers published in this topic that you can easily retrieve from PubMed. And of course, you can download our paper published in the American Journal of Immunology, and among the references, you will find all the scientific studies that support all the statements I have made thus far. In addition to those two microbes and the many others that are in our formulas and that it would take too long to describe, our novel probiotic also contains another molecule, a glycosaminoglycan, a molecule endowed with a number of biological properties that recently has been demonstrated also to perform as a prebiotic. I'm talking about chondroitin sulfate. The evidence for the use of chondroitin sulfate as a supplement in a number of conditions is listed in the official website of the Mayo Clinic, probably the most prestigious medical research institution in the world. Now, it is important to notice, excuse me, that the Mayo Clinic website reports, first of all, that there is no scientific evidence against its use. First of all, do no harm, and the chondroitin sulfate does no harm. And vice versa, there are varying degrees of scientific evidence supporting its use in a number of conditions. Now, the number of conditions that the Mayo Clinic lists where the chondroitin sulfate may prove useful is very, very long. It goes from osteoarthritis to bone diseases, coronary artery disease, eye disorders, interstitial cystitis, psoriasis, and then a long list in alphabetical order, ranging from aging, allergies, Alzheimer's disease, amyotrophic lateral sclerosis, inflammation, inflammatory bowel disease, leukemia, lung cancer, malaria, osteoporosis, Parkinson's disease, down to transplant and wound healing. A very long list that you're free to check on the website of the Mayo Clinic, where chondroitin sulfate may prove useful. Interestingly, an independent study published very recently in January 2017 by an esteemed colleague and friend of mine at the Arizona State University, Professor Jim Adams, shows results that are consistent with our novel concept of a probiotic. Because in this international multi-center study, Adams and colleagues show how the reconstitution of the gut microbiota is associated with significant improvement of autism symptoms. Now, among the very many microbes that constitute the gut microbiota, Adams and colleagues focused their attention on two microbial strains that, according to them, are very important in improving the symptoms of autism. The bifidobacteria that we have already seen and that are highly represented in our formulas, and the Prevotella. Please notice, the Prevotella is not part of our formulas. You will not drink the Prevotella if you drink our probiotics because it isn't there. So you may wonder, if the Prevotella is absent in our probiotic formula, how can it help? How our formulas can help if the Prevotella isn't there? Well, it does help because of the presence of the chondroitin sulfate. As a matter of fact, very recently, it has been demonstrated in August 2016 that chondroitin sulfate is used, we may say it is eaten by the gut microbes as a prebiotic, and it favors, guess whom? The Prevotella. In other words, in the presence of chondroitin sulfate, the Prevotella may proliferate and may colonize our colon, our gut, and reconstitute our gut microbiome. These findings now widen the scope of our novel probiotic concept because not only it enriches the gut and the brain microbiota because of the microbes that it contains that are part of its formula, but in addition to this, that is quite logical, it also favors the balance of other healthy microbes, including those that are not part of the formulas, thanks to the prebiotic effects of the chondroitin sulfate. And chondroitin sulfate also does something else. It modulates the activity of the cells of the immune system, as it has been recently demonstrated in vitro by Dr. Dario Siniscalco and Dr. Annalisa Brigida, who are researchers working at a major European university, who demonstrated that chondroitin sulfate helps establishing the connection between the gut and the brain microbiota because of its effect on the immune system. We're close to the conclusions. We may say that this non-dairy product of ours that has been described in the study published in the American Journal of Immunology and briefly in this talk may truly represent a novelty in the field of probiotic drinks because it combines the healthy properties of the dairy-based products with the properties of the non-dairy probiotic drinks. Now, you may find in your local grocery store many non-dairy probiotic drinks. We feel that our formula shows a number of major improvements over the existing non-dairy probiotic drinks. First of all, the amount of live microbes. Typically, a commercial non-dairy probiotic drink contains about 1 billion of probiotic microbes per serving. The product described here contains more than 9 billion, so almost 10 times as many, probiotic microbes. And this figure derives from the calculation obtained by adding the number of fermenting microbes to the number of non-fermenting microbes, such as the bifidobacteria, and the gastro-resistant probiotics. The presence of the gastro-resistant probiotics is another advantage that you don't find in most, if not all, commercial non-dairy probiotic drinks. Now, gastro-resistant probiotics, which include the bifidobacteria, are microbes that are intended to colonize the gut because they can pass undamaged the stomach barrier. And please notice, most non-dairy probiotic drinks do not contain bifidobacteria at all, neither in their fermenting formulas nor as gastro-resistant probiotics. And in addition to what I've already said about bifidobacteria, it is worth remembering that these microbes are among the first to colonize the human gastrointestinal tract. There is ample consensus that they exert positive health benefits on their host, with particular reference to the immune system, as this paper described in 2016. In other words, the bifidobacteria help reconstituting the healthy core human microbiome, since they are the first that colonize our GI tract when we are born. A third advantage is the presence of eukaryotic microbes endowed with probiotic features, and we have already focused our attention on the Cluiveromyces marxianus. But here I want to give you some further information that may strike some chord. Now, this microbe, in addition to other biological effects that I've already mentioned before, also produces an enzyme that is called lactase. Now, you probably know this enzyme because this enzyme is essential for the complete digestion of all milk, and those of you who are intolerant to milk, maybe they can overcome this intolerance by eating or by having a few tablets of this enzyme that is called lactase. So lactase is an enzyme that is produced by the Cluiveromyces marxianus that is integral part of our formula. Now, for those of you who are interested in applied biochemistry, it may be interesting to remember that lactase belongs to a larger family of enzymes, the so-called beta-galactosidase family of enzymes. And for those of you who are able to read between the lines, it may be interesting to see that according to researchers from the University of Tokushima, Japan, beta-galactosidase treatment is a common first-stage modification of the three major subtypes of GC protein to GCMF, as it was published in this paper in Anti-Cancer Research of 2012. And of course, another advantage over the existing non-dairy probiotic drinks that you may find at your local grocery store is the presence of the chondroitin sulfate, a glycosaminoglycan endowed with a number of healthy properties that now we know it's also a prebiotic that favors the colonization of the gut also by those microbes that are not part of our formulas. This is the last slide. I like this picture very much because it has been produced by the National Cancer Institute of the NIH in Bethesda, Maryland, where I used to work 30-some years ago. And it shows it's an electron microscopy, electron microphotography, showing macrophages actively eating, killing cancer cells. And we hypothesize that this novel product of ours, this novel concept, may find application in all those conditions where you are looking for a natural modulation of the immune system. I thank you so much for your attention. It has been a pleasure to share with you this novel concept of ours. Thank you so much.